Hi everyone. Today I'll be discussing this paper in which authors have talked about how nervous system has evolved in ctenophores in order to understand the evolution of nervous system in metazoa organism. So metazoa are basically multicellular organism, and if you want to understand the evolution, we should look at the evolution of animals in general. So here you can see that the earliest branching lineage of metazoan is ctenophore, followed by Porifera, Placozoa, Cnidarian, and bilateral organism. Now, out of these five clades, only three of them show the occurrence of nervous system, that is ctenophore, Cnidarian, and bilateral. The surprising occurrence of nervous system in ctenophora result in a conflict of the origin of nervous system. So, in order to explain the occurrence of nervous system in ctenophora, two theories were proposed. One is the multiple origin of neurons, according to which there were two events for the origin of neurons, one which took place before the diversification of ctenophora and the second which took place before the bifurcation of cnidarian and bilateral organisms. According to the second theory, there was only single origin of neurons, and somehow during the evolution, the nervous system was lost from porifera and placozoa organisms. In order to understand how which of this theory is correct, which of these theory is correct, and how actually nervous system evolved, how do cell actually communicate to each other, we have to look at the organization of nervous system in ctenophora. So let's talk about ctenophora. How do they look like? So you can see a diagram of ctenophora here. They are characterized by the presence of a mouth, an oboral organ, which is basically an excretory organ. They are characterized by the presence of these comb-like structure, which are lined by many cilia, and they are also characterized by the presence of two long tentacles, which are also lined by cilia. Important to note is that these tiny cilia are very important for the locomotion of ctenophora because they do not have true muscles in them. So these tiny cilia move together in order for the locomotion to take place. You can see clearly in this video, these tiny ripples that you see is the basically motion of tiny cilia, which is facilitating the locomotion of this comb jelly. If we talk about the model organism that they used in this paper, it is Nimiopsis lidi, which is basically a member of Ctenophora clade. And if we talk about the life cycle of the comb jelly, basically they develop from an egg to a larva, which is also called as cydipid stage, followed by further development into the adult. If we talk about the main difference between the larval stage and the adult stage, basically larval stage are characterized by the presence of two tentacles which are lost during the development, and when they become adult they do not have tentacles. If we talk about these two stages, cydipid is the stage that they have used in this paper for all of their experiment because they are smaller in size. So basically an larval stage of comb jelly is around 0.2 millimeter in size. However, once they become adult, they reach a size of few centimeters. So it becomes very difficult to image them in microscope. If we talk about the organization of nervous system, so since these images were drawn in 1991, and from that point of time we have not really developed understanding about the nervous system of ctenophora. So based on the staining of methylene blue, scientists have basically known that they do have neurons, and the neurons do form a nerve net like structure which they call as subepithelial nerve net. And based on what we already knew from metazoan, other terrestrial organisms, it was hypothesized that they will also have axon, dendrite, and everything. However, we really don't know what is the structure of nervous system in ctenophores. So based on these images, it was characterized that they can consist of at least four types of cells. One is called as subepithelial nerve net, second is mesoglial nerve net, and then they have neuronal element at their tentacles and arboreal organs, which are basically now called as sensory cells. So in order to actually understand what is the detailed structure of nervous system in ctenophores or comb jelly, the authors used some fancy technique which is called as serial block phase scanning electron microscopy. So basically it is a modification of scanning electron microscopy, where you take a specimen and you cover it with heavy metal, and then you basically shower electron on the top of the surface of the specimen in order to get the image. In this case, however, the microscope is also characterized by the presence of a diamond knife, which basically acts as a microtome, and the specimen block can move up and down. So basically, once we put the sample inside the microscope will take an image, and the specimen block will move upwards to by a length of 25 nanometer, and then the diamond knife will cut it. And such serial cutting and imaging will take place, so that we will have various images taking place at different z-axis, and when we overlap them we can get a 3D reconstruction of the specimen. So basically authors use this technique in order to get a 3D construction of nervous system of ctenophores or sperm jellies. Now here you can see that they were able to basically make out this very beautiful 3D reconstruction, which shows that coelenteratoid like ctenophores really do have a very elaborate nervous system, which are characterized by the presence of sensory cells and mesoglial neurons and nerve net. Here is a video that they have created. So in the pink color you can see the nerve net, in the yellow color the mesoglial neuron, in the blue color you can see sensory cells, and then you can also see tentacles and other things. But still it is not known how exactly they are connected to each other and how do they communicate with each other. So from our class 12th we have been taught that the neurons are connected to each other via synapses, and that's how they communicate to each other. However, what is the case in this jellyfish in these sperm jellies, we do not know. So here you can see first they looked at the SNN, which is subepithelial nerve net, how they are organized, how they are connected to each other. So this subepithelial nerve net consists of 5 neurons, which are labeled as 1, 2, 3, 4 and 5. And here you can see that via dotted white line the authors have tried to show that when they try to trace back the dendritic processes which are coming off from the neurons, they are actually connected to each other in a continuous form. So basically they are forming a syncytial structure. So in order to further confirm whether they are actually forming a syncytial structure, that means whether they are sharing the same membrane or not, the authors did another imaging experiment where they injected a dye called DII into the two celled embryonic stage of cydipid, and then they later let the comb jelly develop into a cydipid phase, and then they imaged it. So here you can see the two cells basically result in the development of two lobes of this comb jelly. Here you can see that fluorescence is clearly visible on one side of the lobe. However, some fluorescence is also visible on the other side of the lobe. So basically what this dye does, this dye goes and binds to the lipid bilayer, and once it is integrated in the lipid bilayer, it will start glowing. So you can see that where the dye is binding, and this dye cannot actually diffuse through the cell membrane, rather it moves laterally. So only when the membrane is continuous it will able to pass through it, otherwise it will not be able to pass through it. So if we see some glow in the other part of the lobe, then where is it coming from? The dye cannot pass through to the second lobe. So when they did an overlapping of this image with their DIC image, basically they were able to say that the glowing region is actually a part of SNN, that is subepithelial nerve net. So they were able to confidently say that actually the dye is able to cross from this side of SNN to the other side of SNN, because they are actually forming a syncytium. Now, they wanted to understand what is the further structure. Now, surprisingly, even though SNN is forming a syncytium-like structure, that means action potential can directly travel within the SNN by electrical pulses. They do not need to carry vesicles. Still, they saw that these neurons are filled with vesicles which are carrying cargo in it. So in these images, you can see that the yellow dots are basically showing the electron dense region, which shows the occurrence of vesicles which is filled with something. So in their previous paper, which came in 2021, they have already talked about that these ctenophores can produce these ctenophores can produce multiple neuropeptide. They were able to show that they can actually encode for putative 129 neuropeptide, and they were able to show at least four of them are actually existing, like they do get expressed. So they took one of the neuropeptide and created an antibody against it to see if they can actually do immunohistochemistry to show the occurrence of neuropeptide inside these vesicles. And here you can see that the green dots are actually the glowing neuropeptide which are inside these neurons. That shows that these neurons are actually carrying neuropeptide in them. Now the question comes, if and then they further did SDS page in order to confirm the occurrence of these neuropeptide in the neurons, and then the question comes that why do they need to carry vesicles when they are not actually connected to each other by synapses? So later on they actually showed that these neurons are connected, these SNN neurons are connected to other type of cells called as Polster cells, and they form synapses with these type of cells. So in order to pass their signal from this SNN to other type of cell, they do need to form synapses, they do need to carry neuropeptide in their vesicle in order to communicate. Now, in order to understand what is the structure of mesoglial neurons, they looked at their structure in details, and here you can see that there are six mesoglial neurons, and basically if you look closely, these mesoglial neurons are actually, they have their processes parallel to and very close to the neurites which are coming from SNN. And here also in this electron micrograph you can see that nerve net neurons are very close to the mesoglial neurons. So these proposed the idea that they are somehow connected to each other and they are communicating with each other. And later on they were also able to spot some vesicles which are carrying cargo in these neurons also, which suggested that these neurons, the mesoglial neurons, are forming synapses with other type of cells and are able to communicate to other type of cells because of the presence of synapses. Now, because until now we did not have a very good 3D reconstruction or clear uh, understanding of how the nervous system is organized inside the pits, but now that they have very clear images, they were able to characterize different type of neurons. So from their previous paper that I already talked about in 2021, they were able to show that comb jellies have more neurons than we expected. So based on the occurrence of neuropeptide and the expression of neuropeptide in different cells, they were able to characterize 22 putative neuronal type or 22 putative sensory cells. And in this particular study they were able to categorize these 22 sensory cells in 5 classes based on their morphology. Here you can see that the type 1 class consists of a long cilium, long and single cilium. The type 2 structure has short cilium, and also the structure body, the soma body, is also very different from type 1 category. And then type 3 has multiple cilia and they are long, and then fourth category has one long phylopodium, and then finally fifth category has multiple philopodia. Now this is the structural classification, but what can we make out of it? Can we tell how these different type of sensory cells are connected to different SNN or mesoglial cells? Yes. So when they looked at the occurrence or the occurrence of synapses in these different type of neurons, they were able to show that the type 1 neurons or sensory neurons are forming synapses with type 4 neurons, and finally they are communicating signal to nerve net. They also showed that definitely type 4 neurons were able to directly form synapses with nerve net, and because they are connected to nerve net, it is already known that nerve net connect basically help to sense the water vibration and touch so that locomotion can be altered. So basically all of these connection will help in sensing the water vibration to modulate the locomotion of the comb jelly. Then the type 2 sensory cells, they have synapses, they basically form synapses to comb rows, which I already talked that they have comb rows which are marked by cilia, which are also important for locomotion. So they were able to say that basically these type 2 sensory neurons will help in the detection of water flow so that they can fine tune their locomotion. And then finally type 3 time forms synaptic connection to mesoglial neurons, and basically it is known that mesoglial cell basically help in capturing or triggering the food capture. So these functions were assigned to these neurons. However, if you are wondering about what happened to type 5 cells, so even though they were able to like mark the shape correctly, they were not able to find any synaptic connection with any of the cells. They saw that the type 5 sensory cells are in close proximity to polyester cells, which are basically cells of comb rows, but they did not find any synapses, so they have not commented about the function of type 5 cells. Now, finally, to discuss the conclusion or the summary of this paper, in even after looking at all these structural detail, we can't really surely say what is the origin of nervous system. We still do not know whether it is the single origin or independent origin, but it has increased our understanding about nervous system in general. So at this point of time, we always think that nerve system consists of distinct entity of which are called as neurons, and they are connected to each other via synapses. This was actually a question of debate in 1880s. So two of the prominent scientists gave two different theories for the connection of neurons within nervous system. Golgi proposed the reticulate theory, according to which he proposed that all the neurons are connected to each other in the form of syncytium. However, Kajal proposed that neurons are distinct units and they communicate with each other via synapses. And later on, the imaging done by Kajal was able to clearly show that different neurons in birds are connected to each other via the presence of synapses, and they release some vesicles, and that's how they communicate. And hence Golgi's theories were put to rest. However, this study actually shows the importance of this theory, that this is not entirely false theory. We do see occurrence of syncytial nerve net in these calm jellies, and this may be an important point where we can think about the how nerve system actually originated, whether it originated in the form of syncytium, whether it got changed during the time of evolution. But in order to answer these questions, we first need to understand is syncytial SNN is an essential feature for all the member of ctenophores, or it is specific to comb jellies. And also we need to understand what is the status of these SNN and other neurons in the later life history of comb jellies, because all of these experiments were done in the larval stage of comb jellies. But it was like this study is very important turning point, because it opens our mind to different perspective or of evolution of nervous system. Thank you.